Signal transduction, in a simplified version, is the way by which signals out of a cell, rather outside of a cell, are transmitted through specific systems, and thereafter are transmitted into the cell. You can think of this process as an invitation-only party, in which people with an invitation outside of the venue can enter. Those who don't have one cannot. Receptors are molecules located within cell membranes specifically. They are placed there to receive or take in a specific signal, thus ligand, coming from the environment outside of a cell. Think of receptors as a security guard to which you will be handling the exclusive party invitation to. Ligans are the signal molecules that fit into the receptors. Ligans serve the purpose of allowing the receptors to transmit activity inside of the cell. Remember that ligands can be understood by the example of the party invitation and the security guard. The invitation is a ligand, which you are to give to the party's venue security, the receptor, to be able to enter the party, the cell. In speaking of soluble receptors, let it be understood that soluble defines something that can be dissolved. I will focus on a prominent soluble receptor kind, named soluble cytokine receptor. This receptor, on account of activation, plays a significant role in regulating actions within the immune system. Cytokines, as a term, are proteins derived from the immune system that serve as a signaling mechanism between cells to do a certain activity, and therefore in place a form of effect on other cells, hence cell-to-cell communication. Soluble cytokine receptors are receptors that can serve as, quote, cytokine inhibitors, as said by M. Mohini and D.W. Gold in their abstract within the National Center for Biotechnology Information website. Essentially, the soluble cytokine receptor can help as a defense mechanism in immune diseases and other kind of diseases within the body. Soluble receptors can be thought of as the alarm system in the party venue, thus preventing any configuration to happen inside of the cell by firstly preventing what is outside. There are three main classifications of membrane receptors. They are G-protein coupled receptors, enzyme coupled receptors, and ligand-gated ion channel receptors. G-protein coupled receptors are integrated within the cell membrane, with subunits gamma, beta, and alpha beside it. Thereafter, its specified ligand will come into its site. This process will change the shape of the G-protein receptor and will also change the composition of how the subunits are in place. The alpha subunit detracts itself from what it is binding to, GDP, and instead binds to GTP, which is the other factor that changes the place of the subunits. The alpha subunit is moved away from the other subunits, and thereafter each group of subunit searches for target proteins in the cell membrane to come in contact with. In binding to a new protein configuration, the alpha subunit, or the other part of the units, regulates and changes its GDP back for a GDP. This allows it to bind back with the beta-gamma group, and the process continues in repetition. Enzyme-coupled receptors are receptors that simultaneously serve as receptors and as enzymes. Now, enzymes speed up chemical reactions in our bodies, so it is essential to understand this information. This receptor is in two particular physical concepts. When the ligand comes into contact with its two concepts, the receptor is united as one. Keep in mind that up until this point, the receptor is inactive. After this, it is activated. Thereafter, it's unified. The receptor and its subunits begin to bind to proteins and thereafter configure actions within the cell that may or may not be necessary. Lastly, there is ligand-gated ion channel receptors. This receptor is primitive in the nervous system of the human body. Without the ligand in contact with the ligand-gated ion channel receptor, it remains bonded, rather closed, where when the ligand and the receptor come in contact with one another, then is when the receptor opens and creates a pathway for ion components to pass through and be directed out simultaneously. The ions thereafter transform the energy in place within the cell, and transform the cell's actions thereof upon other cells and within itself.